So, welcome back. Today, I am going to talk to you briefly about a very important process in the field of science. In fact, I would say it is the heart of science itself, right? And that is experimental design. And so the whole nature of science, right? If you ask the question, what is science? And science is just a methodological, a certain way of answering a problem. Right? And it's answering a problem in a logical, unbiased, objective way. Right? And so we as humans have a huge amount of innate biases. Right? We have a tendency to want to prove ourselves correct. And in order to find the truth in things, we need a way to isolate ourselves from these biases. We have to find a way to just make sure that everything that we know is actually, to the best of our abilities, true. Okay? And so science is a way that we have gone about trying to achieve this unbiased figuring out of things. And so science relies very heavily upon numerical data, what is called quantifiable data. You need numbers. Right? Good science has numbers. Good science relies upon good experiments. And so today I'm going to talk a little bit about what makes an experiment good versus what makes an experiment bad versus what makes something not even an experiment at all. Okay? And so the starting point for that is the good old-fashioned scientific method. Right? I'm not going to worry about the steps. We're not going to memorize steps, right? But you have to understand the components. Okay? And so scientific method starts off with an observation. You notice something that triggers your curiosity. Okay? And so maybe you notice that all the caterpillars in your neighborhood are purple. And you're like, yo, why are all these caterpillars purple, man? And you want to find out why the caterpillars are purple. Your sister's like, because purple's a pretty color. Right? And you're like, purple is a pretty color, but I don't think that's why the caterpillars are purple. And so then you have to develop a refined research question. And our research question is, why are all the caterpillars in this neighborhood purple? That's it. Simple, straightforward, nothing fancy, to the point. Okay. So we have to come up with some kind of logical, possible explanation for why these caterpillars are purple. And that possible explanation is a hypothesis. And during the next segment, I will delve into what makes a good hypothesis and what makes a bad hypothesis. So we have our working idea. And then we're going to design an experiment that controls the variables around to see if our hypothesis is valid. Right? Do we support our hypothesis? Or do we refute a hypothesis? That's what the experiment does. Okay? So the experiment gives us some numbers to work with. Okay? It gives us data. Love data. Okay? We look at our data, we look at our numbers, and we analyze what the data is telling us. We're not trying to prove our hypothesis. That's bad science. Okay? We're trying to see if the data supports or refutes our hypothesis. And once we have that data analyzed, we come up with a final conclusion. And now in science, nothing should ever be, that's it, we're done. Right? We come to a conclusion and then we extend it. We think, well, let's look deeper into this, right? Is it just this neighborhood's caterpillars that are all purple? How might we use this to help people? How might we use this to help caterpillars? Right? Because maybe all the caterpillars in the world want to be purple. We want to help them become purple caterpillars. Right? So we have to have some extending research after this. We don't just stop figuring it out. We keep on going. Okay? So That's what we're going to look at today. So, one of my favorite things to teach in science is the creation of a hypothesis. And so writing a good hypothesis is what's going to set up your experiment to succeed. Now, a good hypothesis is simply a definitive statement about the relationship between the independent and dependent variable, period. Right? It's a cause and effect relationship. So a good hypothesis about our purple caterpillars, right, might be caterpillars that eat more purple leaves will be more purple. Okay? So I have a cause, eating purple leaves, and I have effect. Caterpillars will be more purple. Okay? Straightforward, no fluff, just right to it. Okay? And now, if I'm designing this experiment, I didn't just pull this out of my butt and throw it against the wall. It's because I went to the neighborhood, right, and I looked around and I made observations. Okay? And I looked at the trees and I said, wow, there's a lot of purple trees in this neighborhood. Okay? Perhaps it's because the caterpillars are eating the purple leaves that they have become purple. And so I've created my hypothesis around that observation. Okay. Working on the same idea, I could create an if-then statement hypothesis, which is the way that a lot of people teach hypotheses, but it's bad hypothesis. That's not how you write hypotheses, right? If you look at scientific literature, you'll never see an if-then statement in a published article. Okay? So do not write if-then statements. Okay? So do not write, if caterpillars eat purple leaves, then they will be more purple. Okay? It achieves the same goal cognitively, okay? But it's just too fluffy. Okay? No if-then. Remove the if, remove the then, and just hit me with a thought. Okay? The cause and effect. Yeah. A much worse, much more terrible, don't ever do it writing way of hypotheses is this bottom one. I think caterpillars are purple because they ate purple leaves. I don't care what you think. Nobody cares what you think, right? Say it with confidence, with determination. This is why it's happening. Okay? And you might be 100% wrong. And guess what? In science, we don't care if it's right or wrong. Right? We don't care if our hypothesis is right or it's wrong. Okay? I could have a hypothesis that's the opposite of this. Caterpillars that eat less purple leaves will be more purple. It's not about being right. It's not about being wrong with your hypothesis, okay? Science is fantastic because it teaches us a huge life lesson. Being wrong is always a first step to being right. Okay? Throwing an idea out there and being wrong is infinitely better than having no idea at all. So never be afraid to be wrong. Okay? Embrace being wrong. It teaches us so many things when we can just be like, yeah, I'm wrong. That's cool. How could I be more right? Okay? So writing a hypothesis is just a definitive statement about cause and effect. So, moving into the actual experimental phase. So we have a hypothesis creating, eating purple leaves will cause caterpillars to be more purple. We have to set up an experiment to test this, to find out if that's a valid statement or not. And so the heart of experiments lie in our variables, right? Things that we are changing on purpose. So we have controlled variables, and we have two of them. We have the independent variable, and that is ultimately the cause. And so in this case, it's the eating of purple leaves. How many purple leaves are we going to feed this caterpillar? And then we have the dependent variable. In this case, it is the how purple will the caterpillar become. So the effect. The independent variable is the variable that you have control over directly. I'm going to feed this caterpillar a certain amount of purple leaves. I control that. I can choose to feed the caterpillar 10 leaves, 8 leaves, 7 leaves, 4,332 leaves, right? The dependent variable is a result of whatever choice I make in the independent variable. I do not control it. Okay? I don't directly make the caterpillar purple. So the dependent variable is the effect. So the independent variable is the one I manipulate. The dependent variable is the one I measure. Okay? Now, these are the only two things that should change in our setup. So if I'm running this experiment on Monday, Tuesday, Wednesday, Thursday, Friday, I want to make sure that my caterpillar on Monday is the same temperature as my caterpillars on Tuesday. I don't want Monday to be a 432-degree day and Tuesday to be a 33-degree day. Because if my caterpillars get more purple on day 2, right, it might be because they changed temperature, or it might be because I fed them more purple leaves. Okay? And so if I have that might be in there, I don't have science anymore. I've lost my science. So I don't know, could be anything. Right? And so everything outside of these two variables, we try to keep the same. Now it's real life, right? You can't control everything in the whole world. You control what you can, and you hope that nothing outside of your control is ruining your findings. Okay? And in order to maintain a certain level of confidence in our findings, we have to use what's called a control group. And our control group ensures that none of that outside stuff is impacting our results. Next topic, control group. So, control groups. Control groups are very, 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 very important to verifying the validity of our data. So when we run an experiment, every single time we do the experiment, we need to run the control group. And so the control group is a group where we have a known relationship between the independent and dependent variable. And so what that means is it's a setup where I know what should happen at the end of it. So in my caterpillar example, right? If I have a regular caterpillar, it's not purple. Regular caterpillars eat regular leaves. So my control group will be feeding the caterpillars 0 purple leaves. Now, theoretically, I know that if I feed the caterpillar 0 purple leaves, the caterpillar should not be purple at the end. So every single time I do my experiment, I have at least one group of caterpillars that have no purple leaves. And now at the end of the experiment, right, I have my control group, they should be not purple. The experimental group that I gave a medium amount of purple leaves might be purple, might not be. I don't know. The caterpillars that I gave only purple leaves, I gave them a lot of purple leaves, they might be purple, they might not be. I don't know. Right? But I do know that the caterpillars who didn't get purple leaves should not be purple. Okay? Now, if these caterpillars turn purple, the ones that go no purple leaves. It shows that these two groups, it didn't matter if I gave them purple leaves or not, because something happened to the caterpillars that would have made them turn purple that was not related to the leaves. Right? So whatever made them turn purple was not part of my experiment. It was an outside factor that I didn't account for. Okay. So my findings in regards to the leaf feeding are just wiped out. They don't matter. It's not a valid experiment. Now, if I have that same setup, right, and my caterpillars who get no purple leaves don't turn purple. My caterpillars who get purple leaves turn purple. And my caterpillars who get a lot of purple leaves turn a lot purple. Right? That indicates that my experiment was valid. Right? Because these guys who didn't get purple leaves didn't turn purple. The control group turned out the way that it was supposed to. So the control group's purpose is to make sure that nothing that you didn't know about messed up your experiment, right? You know with as much certainty as possible that the caterpillars turned purple because of the purple leaves. So that's the importance of the control group. Some people will say the control group works as a comparison group to have a baseline. And no. It's a control group because it makes sure that the experiment is under control. Okay? It makes sure that everything is kosher and good to go. All right? Control groups. So, I've run my experiment on my caterpillars. I've fed some caterpillars a lot of purple leaves. I've fed some caterpillars some purple leaves, and I've fed some caterpillars no purple leaves. And while I was doing that, I was recording my data in a data table. Voila. Okay. And now, as I said at the beginning, for science, it's very important that we have numbers. And now there are some values out there that are more qualitative, right? And so qualitative would be like very red, right? Kind of red, almost blue. Okay? They don't have numbers associated with them. How are you feeling today? I feel good, right? Now, a lot of times we can create scales to apply numbers to qualitative data. It's not as good as having things that are inherently quantitative, right? But it's better than just having like this random scale of good, kind of red. So we want numbers. Now, sometimes things that don't seem like they have numbers actually have numbers. Okay? So for instance, color is one of those things that actually has numbers that you wouldn't think of, right? So there's a kind of red range out there that has a number associated with it. Because light, the colors that you see is light, and it's in waves like this. Right? And so depending on the size of the wave, you get different colors. It's not really the size of the wave, it's the frequency. How bumpy is the wave? Right? So you might have this bumpy wave. This is how bright it is. This is blue, like red, like purple. And so we can measure the frequency, how bumpy it is, and we get a number that relates to color. So over here, I have my amount purple by number. Okay? And I have the amount of leaves by number. Now, if this was a bag table, this would be by mass. Right? So it might be 50 grams of leaves. It might be 75 grams of leaves. Because that's more sciencey, it's better. But for my simplified table, I put number of leaves. Right? Just assume leaves come in uniform number. All leaves are the same. So this is my data outcome. And if I look at this, right, if they ate no leaves, not purple at all. They ate one leaf, not purple at all. They ate two leaves, not purple at all. They ate three leaves, all of a sudden they're a little bit purple. They ate 4 leaves, they're more purple. And they ate 5 leaves, and they're wicked purple. All of a sudden, they're super purple. Right? And so you see a trend in this data. As the amount of leaves increase, the amount of purple increase. This data supports my hypothesis. Right? Supports. Does not prove. Okay? Science doesn't prove things. It supports and it refutes. Okay? This data supports my hypothesis. That is data analysis. And so, once the data has been analyzed, all that is left to do is to create a conclusion. You're going to sum up the findings and suggest the importance of those findings or future research, ideally both. And so the findings of this experiment supported the initial hypothesis that eating purple leaves will cause the caterpillars to turn purple. Further research into other species of caterpillar might provide insights into the mechanisms that cause this transformation. And so sticking on that part about future resources or researchers, future research is very important to science, right? We don't want to just stop here. We want to keep pushing it. Right? And so if you did some chemical analysis on the leaves, you might find that there is a certain purple pigment in that particular leaf that has certain properties that make these caterpillars turn purple. And then you might wonder, well, is it beneficial to humans? Could it turn humans purple? Right? Do humans want to be purple? So these could all be the things you can support or rather suggest in your conclusion. So the conclusion sums up your point and suggests future research. And then that's it. The science is done, and you have figured something else out about caterpillars. Usually you don't do it on purple caterpillars. Usually it's on lots of other things because there's not that many purple caterpillars to research. So yeah, I hope you have gotten something out of this little talk about experimental design. It's something we will talk about at length, ad nauseum, throughout the entirety of the course, because in reality, that's what science is, right? Doing experiments, trying to figure things out in a logical, unbiased way. Yep. So thank you for joining me here at the front of my classroom. See you next time. Have a fantastic day.